Hi, this is Dr. Rosario. So far in this unit, we've discussed the structure of microbial genomes and gene expression. In this video, we are going to talk about the regulation and variation that occurs within microbial genomes. In this video, we have two main learning goals. Our first is for you to be able to explain how genes are regulated, specifically in prokaryotes. We're not going to get into the weeds of eukaryotic gene regulation. It's a little bit more complex, and we're going to focus more here on our bacteria. And what I really want to focus in on is the regulation of transcription. Right? We will talk a little bit about the process that occurs after transcription, but for the most part, we're going to look at how genes are expressed in prokaryotes, how bacteria make the decision which genes are going to be expressed. We're also going to talk about genetic variation. For this video, we're really just going to focus on how genetic variation occurs, but I want you to remember that this will be important as we get later in the semester where we talk about pathogenesis and antibiotic resistance, because both of these things occur as a result of variation within our bacterial genomes. Our last lecture video discussed gene expression. This is the process of using the information stored in DNA to make a functional product. Usually that's a protein. Messenger RNA is produced from a complementary section of DNA during transcription. In turn, this mRNA is used to make proteins during translation. Remember, in our cookbook analogy, genes are like recipes. The cell only reads the recipes that it needs at any given time. What we will be talking about first in this video is how the cell decides which genes to express. There are approximately 4,000 to 5,000 genes on the E. coli chromosome. Not all genes within this bacterium are expressed at one time. That would be energy and resource intensive. In other words, it would be wasteful. This bacterium needs to make only the proteins that are necessary at any given point. Some genes are always expressed. These genes are called constitutive. Examples of constitutive genes include those required for ribosomal synthesis. To maintain homeostasis, a cell is almost always going to need ribosomes for making new proteins. Likewise, components of RNA polymerase are constitutively expressed. Another name for constitutive genes is housekeeping genes. Right? They're necessary for maintaining the house, for maintaining the cell. Other genes are only expressed when needed. Sometimes expression of a gene occurs until a signal indicates that it needs to be turned off. Interactions between signaling molecules, regulatory proteins, and elements in a DNA actually are what result in this gene being turned off. These genes are what we call repressible. Right? They're expressed unless they're turned off. The opposite occurs with inducible genes. These are genes that are not expressed until a signal indicates that they are needed. Before we get into the details of regulation, we must first discuss the arrangement of genes on the prokaryotic chromosome. Remember, a gene is a segment of DNA that encodes the information to build a protein. In prokaryotes, genes are often grouped together by function. These segments are called operons. In this image, you can see a single operon, and there are 3 genes that are found together in this one region of the chromosome. Operons share a promoter and a terminator sequence. The promoter is upstream in a section of DNA called the regulatory region. And the promoter, remember, is the site where RNA polymerase binds and initiates transcription. Also found within this regulatory region, there may be one or more operator sequences. An operator is a segment of DNA that serves as a binding site for regulatory proteins called repressors that will control whether or not transcription will proceed. We'll see that in a moment. Genes within an operon are regulated together. They usually encode proteins with related functions. For example, they may encode a series of proteins needed in a specific metabolic pathway, for the breakdown of a nutrient, or for the biosynthesis of an essential molecule. This allows prokaryotes to regulate these pathways in a single step rather than each enzyme as a separate unit. The mRNA produced in this case is called a polycistronic RNA. It allows for the synthesis of multiple proteins from a single mRNA molecule. By grouping genes together in operons, prokaryotes rapidly adjust to their changing environment and coordinate expression of related genes. Transcription factors are proteins that bind to DNA and regulate expression. There are two types of transcription factors that you should be familiar with here in our prokaryotic cells. Repressor proteins prevent transcription from occurring. Specifically, they bind to operator sites and prevent RNA polymerase from reading the DNA and transcribing RNA. Activators, on the other hand, are the opposite. They enhance transcription. They often bind to the DNA at a site near a promoter and increase the affinity of RNA polymerase to bind and start transcription. Now, so we've laid down some of the basic concepts. Genes are arranged in operons that are co-regulated. You have inducible operons, you have repressible operons. You have repressor proteins that repress transcription, and you have activator proteins that can enhance transcription. Now let's take a look at some examples of operons that are found in E. coli and other bacteria. Tryptophan, you may remember, is an amino acid. E. coli can obtain tryptophan from the environment, or it has the ability to make it itself. The enzymes required for tryptophan biosynthesis are encoded within the TRP operon or TRP operon. The TRP operon is a repressible operon. The cell has a constant need for amino acids for protein synthesis. If there's no tryptophan available, this operon is expressed. The base state for a repressible operon like this one is on. However, if the cell has an ample supply of tryptophan, particularly in the environment outside the cell, it does not need the machinery to make it. The cell needs a way to turn off transcription of the TRP operon. It doesn't need these enzymes. This is where the repressor protein comes in. Repressors are constitutively expressed, meaning they're always present in a cell. In the case of our TRP operon, our repressor protein interacts with tryptophan. Repressor proteins can interact with small molecules that can affect their affinity for DNA. So here in this example, you can see that the tryptophan repressor, there's no tryptophan present, the tryptophan repressor is not bound to the DNA. However, there is a buildup of tryptophan. The tryptophan can bind to the repressor protein, and this increases the affinity of this particular protein for binding to the operator site in this operon. Here you can see that the repressor blocks RNA polymerase from moving down the DNA. Tryptophan, in this example, is a co-repressor. It turns off expression. Expression of the TRP operon is turned off in this scenario. Okay, so the TRP operon is an example of a repressible operon, right? The base state is on, and then the presence of excess tryptophan turns off the TRP operon. The lac operon, on the other hand, is an example of an inducible operon. Lactose is a disaccharide sugar. It is made up of glucose and galactose. It is commonly found in milk products, which is where you may have heard of it, right? So I have to buy Lactaid milk for my 13-year-old because he's lactose intolerant. All right. E. coli has the ability to use lactose as an energy and as a carbon source. 3 genes are required for the metabolism of lactose in E. coli. LAC-Z, LAC-Y, and LAC-A. These genes encode enzymes necessary for the transport and breakdown of lactose. If there's no lactose available, though, the cell does not need to express these genes. The base state for an inducible operon is off. The repressor protein can be seen here bound to the DNA at the operator site. If, however, lactose is added to the growth media, that lactose then can enter into cells and bind to the repressor protein. This causes the protein to change shape and lose its affinity for the DNA. Once the repressor leaves, RNA polymerase is free to transcribe the operon. Lactose acts as an inducer. By adding lactose, we can turn on transcription of the LAC operon. Okay, so this is where I have to get honest with you. The LAC operon is a little bit more complicated than just being turned on if lactose is present. It is actually controlled by more than one transcription factor. In addition to the LAC repressor, there is a protein called the catabolite activator protein, or CAP. CAP works to enhance transcription of the LAC operon under certain conditions. Without CAP, transcription of the LAC operon can occur, but at a relatively low level. That's what we see on the left-hand side of the screen. However, if CAP binds to the DNA, this enhances the ability of RNA polymerase to bind, and that then increases transcription of the operon. This allows for a much higher level of gene expression. So then you might be asking the question, what causes CAP to bind to the DNA? Well, it has to do with cyclic AMP. Cyclic AMP is a molecule that builds up within cells when ATP is in short supply. It acts as an indicator of starvation. Cyclic AMP is what causes CAP to bind to DNA and activate transcription of the LAC operon. Essentially, cells are only going to turn on high levels of expression of this operon if they're starving. So this is a somewhat complicated system of gene regulation. This allows the cells to adapt to different nutrients within their environment. If you grow E. coli in media that contains both glucose and lactose, it will preferentially use up the glucose in the media first. This is because glucose is a better source of energy for the cell. This process of switching from one metabolite to another is called catabolite repression, and it is mediated by CAP. When glucose is available, there's little buildup of cyclic AMP. However, as glucose runs out, the cell begins to starve. CAP becomes activated, and gene expression shifts to the LAC operon. The short lag period you see in the middle of this growth curve is due to the time it takes to produce the proteins for lactose metabolism. Notice that growth continues after this point, but at a slightly slower rate. Again, glucose is a better source of energy than lactose, right? So that better source of energy allows for a faster rate of growth. To summarize, the lac operon will not be expressed if lactose is not present, whether or not there is glucose. In this scenario, the lac repressor is bound and RNA polymerase is blocked. So you can see whether glucose is present or glucose is not present. If there is no lactose, the repressor is bound to the DNA and transcription cannot occur. However, expression of the lac operon is controlled by that second switch. If glucose is present along with lactose, then expression will occur at a very low rate. This is because the activator protein, CAP, is not bound. Maximum expression occurs when there is no glucose and plenty of lactose available for the cells. In this scenario, CAP protein is bound and the lac repressor is not. This maximizes the amount of mRNA that can be made from this operon. Regulating gene expression allows organisms to respond to changes in their environment. So far, the types of regulation that we've discussed has occurred at the level of transcription. While control of transcription in eukaryotes is more complex, it also involves protein transcription factors. There are many other ways that microorganisms can control gene expression. One example is epigenetic regulation, in which the chemical structure of DNA is directly modified, turning genes off or turning genes on. Another is post-transcriptional control, in which there is variable stability of the mRNA transcripts. Right? The more stable an mRNA is, the more it can be used in translation. Finally, proteins, once synthesized, can be activated or inactivated. We see this with our repressor proteins that we just discussed. This is known as post-translational control. While regulating gene expression can affect the phenotype of our microorganisms, the genetic information, the genotype, remains the same. Changes in gene expression are transient. At this point, we're going to turn our attention to how the cookbook, right, the genome of an organism can be altered. That can result in permanent changes. Genetic variation is the differences in the DNA sequence of our microbial genomes. It is the basis for evolutionary change. Organisms with the most advantageous genetic traits are the most likely to survive and reproduce. This is known as natural selection. In the clinical environment, natural selection can drive the emergence of pathogenic strains and the development of antibiotic resistance. Okay, so I have a cartoon here. These are some bacteria, and they're each a little bit different. Now, one of these bacteria has a trait that can allow it to survive under certain conditions. All right, so I'm going to introduce that condition, and I'm going to kill everybody else off except for that one organism. And now what's going to happen is that my bacterial population is now made up of lots of that one organism with that advantageous trait. This is essentially evolution in a nutshell, right? It is change in the makeup of the DNA of a population over time. Okay, so in order to understand genetic variation, we have to talk about the word mutation. This word has a negative connotation, but a mutation is simply a change in the sequence of nucleotides in the genetic material of an organism. It's a change in the DNA. Most mutations are neutral. meaning the change in the DNA sequence doesn't alter the structure or function of a protein, right? It has no effect whatsoever. Occasionally, mutation can provide an advantage. For example, a mutation in a gene for an antibiotic target protein can help a microbe survive under that circumstance where we use that antibiotic. Some mutations are harmful. They result in proteins that don't function properly, and this can provide a disadvantage to an organism. We see this in the case of genetic diseases, for example. Mutations can occur as random mistakes during replication. These are known as spontaneous mutations. Certain chemicals or environmental exposures can increase the probability of a mutation. These are known as mutagens. They can directly cause mutations or increase the likelihood of mutations. The simplest mutation is a change in a single base pair within a DNA sequence. This is known as a base substitution or point mutation. Because of the fact that more than one codon can encode the same amino acid, point mutations are often neutral or silent, and they don't change the amino acid sequence of the final product. In this figure, you can see that a change from an A to a T in the DNA sequence, right there in the middle, does not change the final product of the gene expression. Even though the codon in the mRNA changes from GUA to GUU, the amino acid valine is still placed in the protein at that same location. If a point mutation results in a change in the amino acid sequence, it is called a missense mutation. In this example, a cytosine is changed to an adenine. The protein that results has a threonine in place of proline in the amino acid chain. A single change in the amino acid sequence of a protein can have a major effect on its function. For example, point mutations are known to cause important human diseases, such as cystic fibrosis and sickle cell anemia. Similarly, point mutations can affect the function of bacterial proteins. Nonsense mutations occur when the change in the DNA sequence results in a premature stop codon. Here, a C is changed to a G. This changes the codon from one that corresponds to tyrosine to a stop codon. Instead of producing a fully functional protein, only a partial protein is made. This is a nonsense mutation. Frame shift mutations occur when nucleotides are inserted or deleted from a DNA sequence. Remember that the ribosome reads messenger RNA in groups of 3 nucleotides at a time. Those are our codons, starting with that first AUG in the sequence. If nucleotides are added or removed, it can alter the reading frame or the groupings of those nucleotides into codons. That can completely change the protein sequence. In this example, a thymine and an adenine are removed. Now, where the ribosome once would have added valine to the growing peptide, it adds alanine. Each amino acid after the deletion is also changed. Mutations do not spontaneously occur very often, about once every billion replicated base pairs. Induced mutations occur as a result of exposure to mutagens. 2 major types of mutagens are chemicals and radiation. Many chemical mutagens resemble the structure of normal nitrogenous bases. These are called nucleoside analogs. and they can be incorporated into a new piece of DNA during replication. When that piece of DNA is later replicated, the base pairs don't line up correctly, and a base substitution mutation can result. Another chemical mutagen is one that can insert itself in between the paired bases in our DNA. These are called intercalating agents. This can alter the spacing between base pairs and, again, result in insertions or deletions in DNA that's then later replicated. Several forms of radiation can damage DNA. These damages can increase the likelihood of mutation. Ionizing radiation, such as X-rays and gamma rays, causes the formation of charged atoms or ions that can react with the nucleotides in DNA. This can result in breaks within the backbone of the DNA molecules, either single-strand breaks or double-strand breaks. But it can also alter the chemical structure of our nitrogenous bases, for example, changing cytosine to uracil. Ultraviolet radiation is a lower energy form of radiation. It can cause neighboring thymine bases to form chemical crosslinks called thymine dimers. These dimers must be removed or repaired before these genes can be transcribed or replicated. Often, the repairs that are made result in frame shift or point mutations. In addition to mutation, variation in the genetic material of an organism can occur through what's called horizontal gene transfer and recombination. In horizontal gene transfer, genes are transferred between cells of the same generation. A portion of DNA from a donor cell is obtained by a recipient cell. This piece of DNA then may be incorporated into the recipient's DNA through recombination. There are three types of horizontal gene transfer that occur in prokaryotic cells: transformation, transduction, and conjugation. And we're going to talk through each one of these. Transformation was discovered, somewhat by accident, in the early 20th century by a scientist named Frederick Griffith. He was working to develop a vaccine against Streptococcus pneumoniae. This is a cause of bacterial pneumonia and meningitis. At the time that Griffith was working, there were two common strains of the microorganism. The first, called the S strain, is an encapsulated strain. Remember, a capsule is an organized coating of polysaccharides on the exterior surface of the cell. Often, this can protect a microorganism from destruction by the host immune system. When the S strain is injected into mice, it kills them. The S strain gets its name because the colonies that grow in the laboratory on agar appear smooth in texture, right? That's where the S comes from. The S strain is clearly pathogenic. A second, non-encapsulated strain does not cause disease in mice. This strain is called the R strain because its colonies appear rough in texture. Frederick Griffith experimented with using the R strain as a vaccine against the disease caused by strep pneumoniae. Right? So he injected the R strain into mice. The mice lived, but then if he challenged those same mice with the S strain, the pathogenic strain, they still died. Right? So it wasn't a good vaccine. The R strain was not a good vaccine by itself. So then he tried heat killing the encapsulated strain and using those dead microorganisms as a vaccine. So he heat killed the S strain, injected it into mice. They lived, right, because now that pathogen was dead. Unfortunately, again, the mouse was not protected from infection if it was challenged with the pathogenic strain. Right, so he starts to get creative. Frederick Griffith then tried to mix the heat-killed S strain, right, the heat-killed pathogenic strain, with live unencapsulated bacteria, with live R strain. So he mixed the two together. His results were a bit surprising. All the mice died. It turns out that although the heat-killed bacteria were dead, right, the heat-killed pathogenic bacteria were dead, and the other live bacteria were non-pathogenic, the DNA of those heat-killed bacteria was still relatively intact. What we know now is that the non-pathogenic R strain bacteria took up that DNA from the environment and then incorporated it into its own genome. Frederick Griffith didn't know that it was DNA. He just knew it was something that was transferred from the pathogenic strain to the R strain. He called it a transforming factor. That's where we get this name, transformation. Transformation is the uptake of naked DNA from the environment. This DNA is not packaged inside cells or viral particles. It's just out there in the environment. This process is more likely to occur in bacteria growing under crowded or nutrient-limited conditions, such as that we see in biofilms. Organisms that are capable of transformation are called competent. Some organisms are naturally competent. Turns out that Streptococcus pneumoniae is naturally competent. Once inside the cell, the DNA can then be incorporated into the host genome. This process of adding new DNA or shuffling around genetic information is called recombination. It creates new combinations of genes. In the case of Streptococcus pneumoniae, it gave that non-pathogenic strain the genes necessary for making capsule. Those capsule genes allow that microorganism to then be pathogenic. Transformation is commonly used in the laboratory to create genetically modified organisms. In this case, small pieces of DNA, usually plasmids, are engineered to contain desired genes and introduce them into organisms. There are lots of different laboratory methods used to make cells that are not naturally competent more likely to take up DNA from the environment. This has a wide range of applications. Another way in which DNA can be exchanged between bacterial cells is through conjugation. In this process, a pilus is used to bring two cells close to one another, and DNA is exchanged directly across a bridge in the membranes. Like transformation, the new DNA can be incorporated into the genome of the cell through recombination. The genes for the pili required for conjugation are often encoded on plasmids. Remember, those are small pieces of DNA that are separate from the chromosome. These plasmids also oftentimes will carry the genes necessary for antibiotic resistance. A third method of gene transfer in bacteria is transduction. In transduction, a virus that infects bacterial cells, called a bacteriophage, transfers DNA from one organism to another. So the key thing you want to remember here is that transduction involves viruses. Bacteriophage inject their DNA into host cells. This phage DNA can then be incorporated into the genome of the host and enters a sort of dominant state. Here, we call that a lysogenic phage. This virus can then be copied with each generation of host cells. Sometimes, environmental stress can cause a dormant bacteriophage to become activated. At this point, the viral DNA is cut out of the host genome in a process called excision. Sometimes, a piece of the host genome can be cut out at the same time. This viral DNA is then used to make many copies of the virus. At this stage in the process, it's called a lytic phage because eventually the cell will burst, allowing the new virus particles to be released and infect new cells. Those new cells, however, will receive the viral genome along with a piece of the original host cell genome. This then creates a recombinant organism. One last important element involved in genetic recombination is transposons. Think of transposons as jumping genes. Transposons are small segments of DNA that can move from one region of DNA to another. If they land in the middle of a gene, they can disrupt its function. Each transposon contains the genetic information for their movement, the simplest of which contain a gene for the enzyme transposase, which cuts and reseals DNA, flanked by recognition sites called inverted repeats. Complex transposons carry other genes, such as those for antibiotic resistance. Transposons can be carried on plasmids. They can allow for the movement of antibiotic resistance genes from the plasmid to the chromosome or vice versa. As you can imagine, this can have important implications for the development of antibiotic-resistant strains of bacteria. In this unit, we discuss the principles of microbial genetics. The genes of an organism determine its characteristics. Because they reproduce so rapidly, the characteristics of bacterial populations are constantly changing. Mutation and recombination provide the raw material for evolution. Evolution, remember, is defined as a change in the genetic makeup of a population over time. Organisms with the best characteristics are the ones that are most likely to survive. As we begin to look at the health implications of various microorganisms, it is important for us to remember that every medical intervention can provide selective pressures that can encourage the proliferation or growth of certain traits within bacterial populations. Antibiotic resistance is a prime example.